Hello, everyone. My name is Israel. I am a postdoc at the Unravelling lab at Cornell University. And today I'm excited to be here to talk about the various developmental pathways that shape the complex stem vascular systems in plants. Specifically, today I'll be talking about the evolution of development of stem architectures in the 4 o'clock family, Nyctaginaceae, which was part of my Ph.D. developed at the University of São Paulo in Brazil. I believe we are all fascinated by the astonishing biological diversity of organisms that exists in our planet, right? And understanding how such diversity appeared from the various colors, forms, developments has been one of our primary goals as evolutionary biologists. In plants, one of the topics where we observe an amazing anatomical diversity is on the stem vascular system. For example, there is an important variation in the way that the primary vascular tissues are organized in young stems, constituting the different types of steels. But today, I will focus on the secondary vascular system and the different ontogenies that can produce distinct micromorphologies that we can see in mature stems. The secondary growth of most woody plants is produced by the activity of a single vascular cambium that forms secondary xylem or wood and secondary phloem or inner bark. This is one of the major evolutionary innovations in plants which evolved in the ancestor of the lignophytes. And so among extant plants, the vascular cambium producing that circular structure formed by wood and bark is observed in most seed plants with a few independent loss as we see in the monocots. So the single vascular cambium producing wood and bark is the structure that you can find in most woody plants that are growing around us. But there is a wide range of alternative patterns of secondary growth, which I call vascular variants. And as you can see, vascular variants generate a beautiful anatomical diversity when we look at the stems in cross-section. And these various types of vascular variants, they evolved multiple times across the phylogeny of seed plants. So they are present in gymnosperms, but especially in the angiosperms. And here on the left, you can see summarized 8 types of vascular variants plotted in the phylogeny of angiosperms. Vascular variants are more common in woody vines and lianas, but they are also present in self-supporting plants. And in lianas, they've been considered as an adaptation that maximizes hydraulic conductivity, flexibility, and also mechanical resistance, making these plants able to climb up without breaking apart. So if you are in a trail in a tropical forest and you cut one of these lianas, it is likely that you can find one of these amazing variants that here are illustrated by a Bignoniaceae and also by a Sapindaceae. In terms of origin and development, vascular variants are achieved through the activity of either a single cambium or by multiple cambium. So on the left, you have a lobed stem of a Sapindaceae that was produced due to the differential activity of a single cambium. And in that case, the cambium produced more vascular tissues where we see now the 3 lobes, and less vascular tissue was produced where we see the furrows. On the right, we have another Sapindaceae liana that is showing a stem with successive cambia, which is the presentation of multiple increments of wood and bark in a successive fashion. Well, Nyctaginaceae is one of the families where we can also find vascular variants, and that's why I chose this group to study in my PhD. You probably know this family from the bougainvilleas, also called primavera in Portuguese. Nyctaginaceae includes around 400 species distributed in 31 to 34 genera. They are mostly present in neotropical forests and other types of environments as well. So as a background, I want to show you the phylogeny of Nyctaginaceae, just to mention that the species of this family, they are distributed in 7 tribes. And here I want to highlight 3 of them. Tribe Nyctaginaceae comprise most of the herbaceous plants that grow in arid regions in the US and also in Mexico. Tribe Leucasteriae have most species growing in Brazil and also in Bolivia. And that is the lineage that is sister to the rest of the family. And tribe Caribe has only one species that has been probably extinct. It was endemic from Cuba. So you can also see here that there are different growth forms in Nyctaginaceae, including trees, shrubs, lianas, and also herbs. In terms of anatomy, we know that Nyctaginaceae has some particular features as the presence of medullary bundles that we can see in the pith of this cross-section of a stem of Pisoniella. And these plants, they also have vascular variants. During my PhD, I uncovered the existence of a different type of vascular variant called intersalary phloem. And that was important because all the species of Nyctaginaceae were interpreted as having successive cambia for decades. So following that discovery, two questions that motivated my studies were, how many vascular variants exist in Nyctaginaceae and how did they evolve? And also, do transitions in vascular variants and growth forms impact the diversification rates of the family? So we found that there are four developmental pathways leading to the formation of two vascular variants in Nyctaginaceae. But the first important aspect when we study stem anatomy in Nyctaginaceae is that some lineages start stem development with a regular ustele, and other possess medullary bundles constituting a polycyclic ustele. And there is an enormous diversity of polycyclic ustele in Nyctaginaceae, but this is something for another talk. You can check more information about that in the paper that I cite below. So now let's focus on the complete stem development leading to the two different vascular variants that we found for Nyctaginaceae. The first one is classified as having intersalary phloem, and all the rest have a type of successive cambia. So now let me explain to you the main aspects of each of these ontogenies, and then I'll talk about how they evolved in the family. So the first ontogeny is that one forming intersalary phloem, which is a vascular variant derived from a single cambium. So intersalary phloem in Nyctaginaceae starts with a polycyclic ustele. And next, we have a continuous procambium that produces additional vascular bundles, and that give rise to a new cambium that forms the secondary growth. But the important aspect here is that this cambium presents an irregular activity because it produces secondary xylem and secondary phloem at unequal rates across the stem circumference. And that results in the formation of phloem strains within the secondary xylem. So as you can see, there is a single cambium with a dynamic activity, and that portion that we call overarching cambium is the portion of the cambium that covers the secondary phloem that was formed, and that phloem island become immersed within the secondary xylem. And as that pattern repeats throughout stem development, we have several phloem islands present within the wood. The secondary ontogeny is classified as a type of successive cambia. In this case, stem ontogeny initiates with a regular ustele. So there are no medullary bundles in the pith. However, unlike most woody plants in which a vascular cambium would arise from the procambium of the vascular bundles of the primary vasculature, here we have a case of extrafascicular cambium because that first cambium arises externally to the vascular bundles of that primary vasculature. So after that first vascular cambium produces some amount of vascular tissue, a new cambium arises externally to the vascular tissue produced before, and that repeats several times, and that's why this stem ontogeny is classified as a type of successive cambia. Ontogeny 3 is the most common. It's the most traditional type of successive cambium. So here we have a stem ontogeny that initiates with the regular eustele. There is a period of regular growth from a vascular cambium that forms from the vascular bundles of the primary vasculature. And after some type of secondary growth, we see the first successive cambium being formed from vascular cells because they are found internally to the pericycle and outside the secondary phloem. And after some time that cambium produces vascular tissue, a new cambium is formed outside and multiple cambia are formed. And that's why, again, we characterized this ontogeny as a type of successive cambia. Ontogeny 4 starts with a polycyclic eustele. And here we have a cambium that is originated from the same type of continuous procambium that we saw in ontogeny 1. But in this case, that first cambium has a type of regular activity producing secondary xylem and secondary phloem for a period of time. And only later, a new cambium is also formed from vascular cells located internally to the pericyclic fibers and outside the secondary phloem. Now that we know all the 4 ontogenies for Nyctaginaceae, we can look at how they evolved within the family. So our ancestral state reconstruction showed that ontogeny 1 is ancestral for the family. So ontogeny 2 and ontogeny 3 evolved each once in the tribe Leucastereae that we see in the top, while ontogeny 4 evolved twice in herbs of tribe Nyctaginaceae. Here, it is important to note that successive cambia evolved from interxylary phloem and not the other way around. And this is contrary to our first expectations because, as I mentioned, the literature stated that successive cambia would be inclusive the sole variant that was found in the family. So here we also demonstrate through Pagus correlation test that there is no correlation between habits and the types of vascular variants that we find in Nyctaginaceae. And I also highlight underneath the ontogeny, the phylogeny on the left, that it's interesting to see that there is not a single species with regular anatomy in Nyctaginaceae. All species, all lineages have a type of vascular variant. But we decided to investigate further the correlation of vascular variants and the transitions between growth forms and also the different stem ontogenies. And that's where we started to investigate whether there have been shifts in the diversification rates through time and among lineages. So we found that the evolution of lianas or the climbing habit did not promote diversification of lineages in Nyctaginaceae. So here we performed a diversification rate estimation as implemented in the Bayesian analysis of macroevolutionary mixtures, BAM. And with that, we estimated the net diversification rates, which is the result of speciation minus extinction rates. So our results showed that the most frequent scenario is that occurred 2 shifts in the diversification rates in Nyctaginaceae. One encompasses the tribes Pisoni, Bougainvillea, and Nyctaginaceae, and the other one is found in the genus Comicarpus. The most interesting aspect to observe is that the shift that we see in diversification rates across the phylogeny of Nyctaginaceae, they do not coincide with the evolution of the climbing habits in the family, which are indicated by the arrows that you see in the right. So this is interesting because it shows for the first time with a specific analysis that being a liana or a climbing plant did not promote diversification of the lineages. And that is contrary to the hypothesis that the evolution of the climbing habit promotes diversification as stated by Guillenol in 1994 or 2004, excuse me. Therefore, at least in the context of Nyctaginaceae, where we find vascular variants present in all lineages, these hypotheses from Guillenol did not hold true. And because of that, we asked if vascular variants or other anatomical features would be a characteristic impacting the diversification rates in Nyctaginaceae. So we decided to evaluate the contribution of morphological characters in the diversification of Nyctaginaceae. And for that, we applied a trait-dependent analysis using the hidden state speciation and extinction method, HIS, for 3 characters. So we evaluated habit, comparing self-supporting versus climbing plants, the primary growth, comparing regular eustele and polycyclic eustele, and secondary growth, comparing regular versus vascular variants. And by applying these model comparisons, we found that only the eustele types relates with diversification rates in Nyctaginaceae, as the acquisition of the polycyclic eustele was associated to elevated rates compared to the regular eustele that we see in purple. So it seems that neither habits nor the transitions of vascular variants were associated with higher diversification rates in Nyctaginaceae. And given these interesting results, I'm now investigating the contribution of medullary bundles to the form, function, evolution, and diversification of Caryophyllales, the whole order, because medullary bundles evolved also multiple times in this group, and they seem to have interesting form-function correlations and perhaps with species diversification as well. So I want to thank the conference, all the organizers, this conference and also the symposium. I thank all the institutions and the people involved in my research for my PhD, and I also thank Cornell University and the Unravelling Lab for supporting my research in the present moment. So thank you so much.